It's important that we talk about technology in our biopharma investing practice this year because over the last decade or so, several revolutionary new technology platforms have emerged which are fundamentally changing our approach to treating and curing a number of diseases.
Well, I've been in the industry now 35 years. So when I first started, it was little white pills for all sorts of diseases, whether it's depression or asthma or ulcer disease. Gene and cell therapy used to be science fiction. Well, today they're here. There are approved gene therapy drugs available. Cell therapy, the cell therapies, before you could have never thought about actually having a cell engineered to produce proteins that would be therapeutic, and that's what's going on right now in the research labs.
When we talk about cell therapy today, typically what we're referring to is using genetic engineering technologies to engineer cells, human cells, to make them more effective when re-administered to a patient at fighting disease. I love Battersea because it really comes out of the hard work that Ali has done over the last 3 to 5 years scanning all the current technologies in cell therapy.
So when we first started looking at the space, we really had seen a lot of the shortcomings that are now becoming apparent with current cell therapies. And so one of the things that we were trying to do is figure out how do we make these cell therapies better. And so we formed this company, Battersea, which essentially the goal of it is to create the next generation cell therapy company. Yes, so T cells, the reason people have focused on them, that's where the data is today. That's where the breakthrough has been made. But there are a whole host of other immune cells in your body. And so, part of the question is how do you harness those other cells? And so certainly now we are at Battersea starting to look at other types of cells like natural killer cells, for example. Another way is trying to improve upon the current T cell platform. So, today the current cell therapy platforms are really targeted at finding one specific tumor marker. And that works great. But I almost liken it to starting a fire. The T cell will do what it's supposed to do, which is go and find a cell that has that specific tumor antigen. And when it does, it kills it. And so the idea behind going after multiple antigens is if you can start a much bigger fire that can catch on its own, then you can get a broader immune response and hopefully more durable responses.
We all remember back around the year 2000 when we sequenced the human genome, and the lay press wrote about now the ability to cure all sorts of previously untreatable and incurable diseases. In fact, one of our own partners, Jim Barrett, was really one of the fathers of gene therapy. In reality, it's taken us 15, 20 years to learn how to apply what we learned through the sequencing of the human genome to actual drug development and to make human disease better.
So Nightstar is an ocular gene therapy company, and we're focused on inherited retinal diseases that cause blindness. So viruses do one thing exquisitely well. They infect tissues. So we insert the gene into the virus, and then we insert that virus into the eye with the express purpose of infecting tissue and allowing the gene to get into cells. And then ultimately that gene will produce a protein that will restore function.
The eye is a really attractive place to work in gene therapy for a number of reasons. Interestingly enough, there are literally hundreds of rare genetic diseases of the eye, many of which lead to blindness in early to midlife for patients.
We're working on a range of inherited retinal diseases. Our first one's in choroideremia, which is a blinding condition with no cure today. I've been with a lot of patients and parents, and it's incredibly emotional disease because if you can imagine, they're born with good vision, they're diagnosed with this disease, and they're told it's a one-way ticket to blindness. There's really nothing we can do about it. What we've seen now in 32 patients that we've dosed is quite a significant effect on their vision. So what we're seeing is maintenance of vision in the treated eye while the untreated eye gets worse. We have a subset of those patients, though, who've had a hyper response, meaning a significant improvement in visual acuity. So we have, out of the patients that we've treated, we've got about 20% of those that have had a 3-line improvement on a Snellen chart, which is really remarkable. We finished our phase 1/2, which is to establish essentially safety and dose. And our plan is to move into phase 3 in the beginning of 2018. We've already begun dosing patients in the second program, and it's for retinitis pigmentosa. We have a third program, the Best's disease, should be in the clinic by 2019 with that program. So we'll have a really nice runway of programs.
What we're seeing today is that technology is able to be applied to drug development and to treatment of disease much, much faster. The discoveries and innovations that led to CRISPR technology and gene editing are only a few years old.
So gene editing, at the heart of it, is trying to look for areas in your DNA where there are mutations and cutting those mutations out and correcting that mutation so that the defective gene isn't being expressed.
CRISPR Therapeutics is a company founded based on a revolutionary gene editing technology called CRISPR. CRISPR technology can be used to permanently make edits to the human genome and thus treat hundreds of different diseases.
A few years ago, the idea of correcting your mutations at the DNA level were no one could really contemplate, but with CRISPR-Cas9, that's becoming more of a reality.
CRISPR is a molecular mechanism that was developed within bacteria and honed over millions of years to help protect it against viral attacks or phage attacks. The CRISPR technology uses DNA-RNA base pairing, where a small guide RNA with a 20 base pair sequence essentially serves as a zip code that tells the CRISPR molecular scissors where to cut in the genome.
The idea of being able to correct a defect that's in your DNA that you normally couldn't with these molecular scissors is an amazing concept.
Yeah, I think the important thing to understand about CRISPR-based therapeutics are that they're one-time and they can be curative. Imagine kids with sickle cell anemia or beta thalassemia. You have one intervention early in life where you put these edited cells back into the body of the patient and they're cured for life. While the early applications of CRISPR are focused on defined diseases with defined mutations, the applicability of CRISPR is wide. The more we understand the different mutations and the different patient subsets that have the different mutations, the more effective therapies we can develop using CRISPR. It's a once-in-a-generation, if not a once-in-a-lifetime type of platform that we also believe will mature quite quickly because it's coming at the same time as the confluence of human genetics and the maturation of delivery technologies.
So I know you'll have heard about Battersea and CRISPR. Those are examples where cellular engineering is taking place. It's not too dissimilar to what Synlogic is doing, except that the cellular engineering they're engineering is around bacteria.
Synthetic biology is really fascinating, and it's a terrific intersection between our technology practice and our healthcare practice, in that what we're really doing is we're programming cells.
As in Logic, we engineer probiotics, the same probiotics that you buy in Whole Foods or in a health food store. What we do is we engineer them genetically. We change the genes inside to make them to perform a metabolic function that you cannot make with your own body. And we do this because it turns out that we have a very interesting organ that is a bacterial organ in our body called the microbiome. And that organ that we didn't study in medical school is sending all the time signals in our blood. In fact, if we check our blood right now, 100 or 200 metabolites in our blood are coming from that highly metabolic organ that is not human, is bacterial. And very likely, the difference between health and disease in many states, in metabolic disease, in immune disease, probably has some relationship with that organ itself. What we do is we add a function to that organ, and it's the function that you are missing in your own body. A key component of our technology is what we call switches. And these are elements that are like turn on or off the therapeutic circuit that we have built. And not only turn on or off, but they're coupled to a sensor. And the sensor is able to determine if you have certain pathological state. We always use the same probiotic bacteria. And then we change the genetic program disease by disease. It's like an iPhone. We always have the same iPhone, but the same way that when you open an app, it's now a music device, or you open another app, is now a camera. Same thing happened for us, completely different function. The speed and the precision is unlike anything that I experienced before. We have 4 indications that will go one by one that is dealing with a different metabolite, a different app, a different genetic circuit, but the same concept. That is, I think, the tip of the iceberg of our pipeline.
So they've gone from moving into the lab to filing an IND in 3 years, which you never hear of in biologics or in the small molecule world. As we get more and more advanced with the types of therapies, whether it's cell therapy or gene therapy, and in here, I mean, the benefit is the agency already knows about probiotics. They're out there, millions of people use them. The only thing we're doing is we're engineering certain different features to them. So from a safety point of view, we've already covered that hurdle.
The beauty of gene therapy or gene editing or cell therapy is you're actually trying to engineer the cell to behave a certain way. And so it's really a new paradigm in terms of how do you create a new novel drug.
So it's the tremendous depth and breadth of our investing team and the way we all work together that we think is enabling us to identify the best and most investable opportunities in these revolutionary new technologies. In the cases of 3 of the 4 companies that we're really focused on today, NEA has been intimately involved in the very creation of each of these companies, from either the seed round or the Series A round. It's really interesting to see the evolution in our practice where we're now playing such a strong leadership role in the creation, ideation of these early stage companies.